This topic lecture covers microtubules. Here are the objectives for this topic. In this lecture, we are going to discuss microtubules a little more than we will discuss the other two components of the cytoskeleton. This is because microtubules have more components to them and participate in a variety of functions within the cell. Unlike the rope-like structure of intermediate filaments, microtubules actually form a tube-like structure. This structure is composed of 13 protofilaments. These protofilaments are linear chains of alternating alpha and beta subunits. The 13 protofilaments non-covalently bond to each other to form these tubules. One very important aspect of these microtubules is the polarity of the tubulin. When the beta subunit is exposed at the end of the microtubules, this is said to be the plus end. Conversely, when the alpha subunits are exposed, this is said to be the minus end. Where do these microtubules come from? They come from organelles known as centrosomes. These centrosomes are found only in animals and contain two structures that look like churros. These are known as centrioles. We aren't quite sure what their role is in the cell yet because they do not participate in the production of the microtubules. The microtubules are instead grown out of a third subunit of tubulin known as the gamma subunit. This site is known as the nucleation site. Let's now take a look at how these microtubules are assembled and disassembled. These processes are best viewed as a video, so I encourage you to check out the videos that I have posted for you in Blackboard. Microtubules can grow and shrink. This is how they pull the chromosomes apart during cell division. The growth of the microtubule will always proceed from the plus end of the microtubule or the end that has exposed beta subunits. This process can be quickly reversed, and the assembly and disassembly is known as dynamic instability. This instability has to do with how the process happens. The growing of the microtubules requires energy in the form of GTP, but they are added so quickly that the GTP isn't hydrolyzed with this. This makes a section of the microtubules that have GTP incorporated into the microtubule. When these GTPs randomly hydrolyze, this can reverse the process and cause the microtubules to disassemble quickly. This process is continually balanced within the cell. The centrosome will continually cast microtubules out and pull them back in. The only activity in the cell that prevents the microtubule disassembly is when the microtubules bind to another structure within the cell. The ability to prevent microtubules from forming or disassembling is an interesting drug target within the cell, and your textbook goes into some details in this if you are interested. Now that we have covered what make up tubules and how they assemble and disassemble, let's talk about what these microtubules do within the cell. They have two main functions. These are to help with intracellular transport and to help provide structure to larger structures involved in cellular movement. Let's look at each of these in depth. In Biology 1, you learned about how the cell uses microtubules to pull apart the chromosomes, but we haven't talked about how the cell can move items around the cell. We talked about vesicle budding and how vesicles move from one organelle to the other, but how do they get there? They get there by utilizing motor proteins that walk along the microtubule. There are two classes of motor proteins, dynin and kinesin. These motor proteins move in opposite directions on the microtubules. Motor proteins not only help move small items, they can attach to organelles such as the Golgi and ER, ensuring that they are stationed in the correct locations within the cell. These little motor proteins are pretty cool, so you should check out the videos on Blackboard when you get a chance. Another role microtubules play in the cytoskeleton is through the added stability to cilia and flagella. Let's talk about these structures. Cilia are hair-like structures that stick out of the cell. They are still covered in plasma membrane, so they are an internal part of the cell. These structures can propel cells through their environment or move substances over their surface to either collect nutrients or pass items across the cells. Ciliated epithelial cells are an example of cilia being used to move substances over their surface. Flagella also help cells move through the environment and tend to be much longer than cilia. The classic example of a flagellum is the tail of the sperm. Microtubules make up the central shaft of both cilia and flagella and are arranged in a 9+2 format. There are 9 microtubules that make up the outer structure. Then there are 2 microtubules that form the center of the structure. The tubules produce movement by bending at the core to make the whip-like action. Other proteins are utilized to help with this bending movement. One main protein that helps are the dynon arms. These help the microtubules slide next to each other in the bending motion. This is the end of this topic lecture on microtubules. Please review the objectives for this topic and let me know if you have any questions.